Okay, so as in the last video, we have discussed about nucleus and nuclear envelope, which was the covering. Here we're gonna discuss about the nuclear pores, right? Yeah. Here we are going to discuss about nuclear pores. Second part. Fine. So nuclear pores are basically these help in communication between the cytosol and nucleoplasm. As nucleoplasm is a fluid which is present inside the nucleus and outside the nucleus we have cytoplasm. And I taught you that cytoplasm have two parts. One is cytosol and another is cytogel. So here I'm going to draw the diagram. As here we have fluid present. Sorry, let me highlight. Here we have fluid present. That fluid is known as cytosol. That fluid is known as cytosol. While here at the end of this membrane we have another fluid that is known as cytogel. That is known as cytogel. However, mm, inside the nucleus we have fluid present that is known as nucleoplasm. Nucleoplasm. So cytogel is the basically the fluid which is present near to the plasma membrane and which is more viscous. बहुत गाढ़ा होता है. So mostly the organelles are present in the cytosol. So the connection between cytosol to the nuclear plasm, that connection is done through the pores which are present at the nuclear membrane. Those are nuclear pores like this. Like okay, hopefully this diagram is clear to you a little bit. Now here from the nuclear pores, what actually move inside and outside. So as we know, for the DNA transcription, RNA transcription, rRNA, tRNA, these are the proteins. Oh, sorry, these are the rRNA genetic material which move plasm by the nuclear pores, move in cytoplasm. From these are present in the nucleus, which move in cytoplasm by nuclear pores. Okay, so here the active and passive transport both will occur. Some material will move by the actively and some will however move by the passive. So both active and passive both will take place. Fine. After that here this was the second one. Now we're gonna discuss third part, that is nucleoplasm. Here I'm gonna discuss our third part, that is nucleoplasm. Now as I told you what actually this nucleoplasm is, so this is basically this fluid present in the nucleus that is known as nucleoplasm. Fluid present in the nucleus. However, that fluid present in the nucleus is known as nucleoplasm, or that is known as karyolymph. That is known as karyolymph. Here karyo word is used for the nucleus. Karyo word is used for the nucleus. However, lymph is used for the fluid or colorless fluid. Fluid, colorless or transparent fluid. Okay, so ट्रांसपेरेंट सेमी फ्लूइड कंटेन, जो इसके अंदर ट्रांसपेरेंट सेमी फ्लूइड जो प्रेजेंट होता है, विच कंटेन डीएनए, आरएनए, न्यूक्लियोप्रोटीन, like this, all the material which is present. So this, let me write here, transparent semi fluid. Fluid contain what? It contain DNA, RNA, and nucleoprotein. Nucleoprotein. Okay. After that, these nucleoprotein include what? Now, the nucleoprotein क्या है? So let's discuss this nucleoprotein. Nucleoprotein are basically DNA plus stone. DNA plus Histone protein are called as nucleoprotein. Now here DNA have negative charge, however histone protein have positive charge. Just remind this. Okay. Histone protein are basically basic protein which are rich in lysine. Lysine is one type of substance we can say, yeah. Now here this nucleoplasm contain various enzymes for replication, transcription, post-transcriptional modification and RNA transportation like this. So we can say this contain enzymes, multiple enzymes. Okay. After that here we have chromatin network contain DNA plus histone protein. Now here in the nucleoplasm के अंदर एक हमारे पास है ना chromatin धागेनुमा structure which is colloid structure is present. That structure contain what? That structure contain DNA plus histone protein. Chromatin, a thread-like structure. Colloid structure. Thread-like colloid structure which contain what? Which contain DNA plus protein, that is histone protein. Like now this chromatin is divided into two types. Chromatin is divided into two types. Number one type is heterochromatin. Another we have euchromatin. Heterochromatin and euchromatin. Fine. Now what actually this heterochromatin is? As heterochromatin is a region which is directly stained region, जो कि क्या है? Dark region है. Darkly stained region is present. However, this is loosely or lightly stained region. Lightly stained region. Now here in the heterochromatin, genes are tightly declared. Means genes tightly एक दूसरे के बाद declare हो जाते हैं. Genes are tightly declared. However, in the euchromatin, genes are not tightly declared, these are loosely declared. Fine. And in the heterochromatin, transcriptionally inactive genes are present. They do not form the RNA. Transcriptionally inactive genes are present. Which do not form RNA. However, here these transcriptionally genes are active and these contain RNA too. Fine. So this was all about the nucleopore and nucleoplasm. Hope you get something new from this video. In the next video, we're gonna discuss about the chromatin network and nucleolus. Bye bye.